would like to move to the next presentation. So Professor Koji Fukui, please.
Thank you very much for your kind introduction. So my name is Koji Fukui. I come from Saitama Prefecture. So I am in Shibaura Institute of Technology. So that is a very small private university in Japan. So today I would like to talk about the basic research about the measurement method of ESR on using Twendee X. I bought ESR machine last year, so today is a first presentation about my laboratory data. So there are a lot of disease deeply related to the ROS. So as you can see, very famous many disorders are deeply related to the onset or progression of disease. So reactive oxygen species are produced from mitochondrial respiration systems. So very high energy electron bind to oxygen and produce singlet oxygen, and gradually produced superoxide and hydrogen peroxide and hydroxyl radical. So there are a lot of reactive oxygen species produced in mitochondria. So there are a lot of presentation in this meeting. So many researchers presented about the relationship between the aging and the ROS reaction. So accumulation of oxidative damage accelerates pathological aging. It is not physiological aging. So the balance of the production of reactive oxygen species and the volume of the antioxidant substances are very important. So to keep the balance is very important to maintain for our health. So Twendee X is developed by Professor Haruhiko Inusa in 2005. So Twendee X include 8 beneficial substances, so such as coenzyme Q10 and vitamin C and others. So there are a lot of scientific papers. So treatment with Twendee X is a very good beneficial effect for our health using in vivo and in vitro and human and animal experiments. So today's my presentation is about the measurement of radical scavenging activity of blood using animal model after treatment with Twendee X and others. So basically, to measurement of oxidative stress condition, we have a lot of measurement methods, so such as measurement of antioxidant substances volume and measurement of antioxidant enzyme activities and oxidative products such as lipid hydroperoxide and 808DZ. But these indexes are indirect method, right? So if we will measure these indexes, we always use HPLC and PCR and other machine. So I want to show about the basic data about the ESR machines. So In the case of ESR, so to mix with spin trap agent and samples and directly detect the reactive oxygen species. It's okay. So, yeah. So this is a example of a measurement of superoxide. So we mixed with antioxidant vitamin. So that is ascorbic acid, and mixed with antioxidant vitamin, ascorbic acid, and spin trap agent. So as you can see, ESR spectrum showed gradually decreased in ascorbic acid concentration dependent manner. So treatment with 1 millimolar ascorbic acid, superoxide spectra of ESR completely disappointed. Next, we checked about the measurement of hydroxyl radical. We use the same antioxidant vitamin, so that is ascorbic acid. So treatment with ascorbic acid gradually decreased the hydroxyl radical ESR spectra in a concentration-dependent manner. Next we try to the radical scavenging activity of Twendee X, and we mixed with Twendee X and spin trap agent in a test tube and checked about the radical scavenging activity by ESR. So as you can see, treatment with Twendee X gradually decreased the volume of superoxide. So the volume of Twendee X shows a positive correlation with decreased volume of a superoxide amount. So next, we tried to the same experiment, but this experiment we measured by a hydroxyl radical. So as you can see, treatment with Twendee X gradually decreased hydroxyl radical ESR spectra. So this result also showed that positive correlation decreased volume of hydroxyl radical and the treatment with Twendee X volume. So finally, we checked about the peroxyl radical. So as you can see, treatment with Twendee X significantly decreased the radical spectra on ESR. So Twendee X showed a very strong radical scavenging activity. As a next step, so it is necessary to clarify whether administration of Twendee X affect free radical scavenging in the body. So our next experiment is a preliminary experiment. So we try to detect of the radical scavenging activity in serum. So in the case of our experiment, we always use animal because I'm not a medical doctor, so I took blood from the mice and separate serum and divided to the 3 different dilution range and checked about the radical scavenging ability. So as you can see, the amount of superoxide did not dramatically change in 3 different dilution range in the serum. So next, we checked about the difference of storage period. Because in the case of human blood sample, each sample different frozen storage period. So it is very difficult to whether every indexes are correct or not. Okay, so take a blood, separate serum, and divide it 3 different new tubes. So one sample is a no frozen sample. So other 2 samples, put it on a deep freezer, and one is a 1 week frozen storage, and the next one is a 2 weeks. So we compared about the radical scavenging activity by using a ESR machine. So as you can see, so non-frozen sample and 1 week frozen and 2 weeks frozen, the result of the superoxide amount did not dramatically change. So this result indicated that 2 weeks frozen sample can use measurement of radical scavenging activity. Okay, but this experiment, we used serum sample. Before starting of this experiment, we separate from the whole blood. But serum, serum is a very big loss volume. It takes about 30% or 40% loss. So sample volume dramatically decreased compared to the whole blood sample. In my future plan to elucidate the antioxidant activity of Twendee X by ESR, blood must be collected from the same mice at different time from the tail. So in the case of tail, it is very difficult to take a large amount of blood. So next experiment, we try to measure the radical scavenging activity by using plasma. So to compare the volume between the serum and the plasma, we can get a large volume compared to the serum. So this is a non-frozen plasma sample. Before start of experiment, we checked about the heparin. So as you can see, the orange line shows ESR spectra did not differ between the only PBS sample and the heparin sample. So it was no influence in the presence of heparin in a plasma sample. And next, we measure the radical scavenging activity of superoxide. So we made different dilution range and checked about the ESR spectra and gradually decreased. So this result indicated that plasma sample can measure the radical scavenging activity. So now we are continue to study other free radical scavenging activity and frozen plasma samples. So before next experiment, we made a plasma and serum by using from the same mice whole blood. And we made a plasma and serum and compare the result of radical scavenging activity. So as you can see, the ESR spectra did not differ between in plasma and serum. So this result suggests that the plasma sample can measure the radical scavenging activity. Finally, we tried to the radical scavenging activity and compare to the edarabone. So orange circle shows the result of the edarabone, and blue circle shows the result of the blue circle shows the result of Twendee X. Blue is Twendee X. Okay, so Twendee X is effective at low concentration compared to the edarabone. So hydroxyl radical scavenging activity and superoxide radical scavenging activity, both radical scavenging activity are higher compared to the edarabone. So this is a conclusion of my presentation. So Twendee X was found to high free radical scavenging activity such as superoxide and hydroxyl radical and others. A method for measuring free radical scavenging activity in blood sample, especially plasma, was established, and frozen sample. So Twendee X had a higher free radical scavenging activity than edarabones. So future plans, so investigate the duration of Twendee X free radical scavenging effect. So we continue to study about this experiment, and we will be able to develop a custom-made supplement. Thank you very much.